Hello and welcome to another tutorial for BIOS E1A. In this tutorial, we'll walk through a practice problem that's all about polymerase chain reaction, or PCR. Before we get to figuring out the next nucleotide synthesized during PCR in Part A of the problem, and before we get to picking the matching primer in Part B, let's first review how PCR works and what you guys learned about PCR from lecture. To do PCR, first you need a DNA template. When you're performing PCR, the goal is to amplify a particular region of your DNA template. In other words, to make many copies of that region of double-stranded DNA sequence. In order to make those copies, in addition to your DNA template, you also need primers to do PCR. PCR primers are short DNA oligonucleotides. That is, they are short sequences of single-stranded DNA. In reality, they are usually at least 18 to 20 nucleotides long. But for the purposes of this example and other course practice problems, we'll usually use shorter example primers that are only a few nucleotides long. A single primer isn't enough to amplify your region of interest by PCR. You actually need two primers. A pair of primers is needed to amplify a sequence by PCR. The crucial information to remember about PCR primers is that they will match the sequence at each end of the region you will amplify. Specifically, the primers will have the same 5' to 3' sequence as the DNA template at the two 5' ends of the region you're amplifying. You can see that with the example here. Primer number 1 has the exact same sequence, 5' to 3', as the top strand does at the 5' end of the amplified region. For primer number 2, you'll need to read right to left to see it here, but primer number 2 has the exact same sequence, 5' to 3', as the lower DNA strand. Again, keeping in mind our light blue box of region we want to copy here, this primer will be at the other end of the region you want to amplify by PCR. This will make a little more sense after we walk through an example of PCR cycles. To carry out PCR, you need four main ingredients. The first is a DNA template. The second is many copies of your two PCR primers. Then you also need the dNTP monomers that will be used to synthesize the new DNA copies. And finally, you also need the protein enzyme that will actually do the DNA synthesis. This enzyme is one you are already well acquainted with from Lecture 2.1, namely DNA polymerase. For PCR, laboratory researchers typically use special versions of DNA polymerase that can function even after multiple rounds of heating to higher temperatures. Researchers use DNA polymerases for PCR based off of the DNA polymerase proteins isolated from thermophilic bacteria that live in hot springs and hydrothermal vents, where they'll need a DNA polymerase enzyme that's more thermostable and capable of surviving heating. The first thermophilic bacterium whose DNA polymerase was isolated for use in PCR was called Thermus aquaticus, which is why the DNA polymerase enzyme used in PCR is often referred to as Taq polymerase, or just Taq for short. Why would using a thermostable DNA polymerase be so desirable for doing PCR, though? Well, that makes much more sense when you review the actual steps of the polymerase chain reaction. PCR is accomplished by cycling your reaction mixtures through the same three steps, typically 30 or more times. The first step is the denaturation step. It involves heating the reaction mixture to near boiling, about 95 degrees Celsius, in order to denature your DNA template. That is, separate it from a double-stranded molecule into two single-stranded molecules. Then you proceed to an annealing step, where the reaction is cooled to usually around 55 to 65 degrees Celsius. In this step, your primers anneal or bind to the DNA. Third, there is the extension step, which occurs at around 72 degrees Celsius, at the optimal temperature for the polymerase enzyme to function and extend the newly synthesized DNA strand. Let's walk through the first few cycles of PCR to see exactly what this means. Let's start at the beginning, with cycle 1. Here's our DNA template. I've marked off the ends of the region we want to amplify with the nucleotide sequences in white. Remember, these sequences at the 5' boundaries of the region you're copying will have the same sequences as your primers. Next, the first step of cycle 1 of PCR will be denaturation. The reaction mixture will heat to 95 degrees, which breaks the base pairing hydrogen bonds in double-stranded DNA and opens it into two single strands, which primers can bind to. Then the temperature is lowered and we go to step 2, the annealing step. This is where the primers we've added to the reaction mixture do their part. As you can see here, again, the primer sequences are the same as the sequences in white that I've left showing on the purple DNA template. This is because the primer sequence will match the sequences marking off the ends of the region in the DNA you want to copy. Your PCR primers will base pair with their complementary sequence on the other strand, across from the sequence that they are identical to. Then you'll proceed to step 3 of the first PCR cycle, the extension step. You'll ramp up the temperature for step 3 just a little bit, to usually 72 degrees Celsius. This is the ideal temperature for DNA polymerase in your PCR mix to function. It will synthesize new DNA strands, just like you learned about in Lecture 2.1 for DNA polymerase, growing those new strands off of a primer and growing them out in always the 5' to 3' direction, which is marked by the green arrows here. We're doing this reaction in a test tube, and we're controlling which primers are there, namely just our 2 PCR primers. So DNA polymerase will do its thing during this extension step and polymerize new DNA strands off of just our PCR primers. And at this point, we finished the first cycle of PCR. It's called the polymerase chain reaction, though, so we keep on cycling. The next cycle begins again by raising the temperature up to 95 degrees, nearly boiling, and doing the denaturation step. The purpose of this step, again, is to separate the double-stranded DNA into single strands, so that when we go to step 2 of the cycle in the annealing step, our PCR primers can anneal or bind to the single-stranded DNA. Then we go to step 3, the extension step. DNA polymerase will do what DNA polymerase does, and make new DNA strands in the 5' to 3' direction off of existing primers that we annealed in the previous step. Now we've finished 2 cycles of PCR. Instead of one original double-stranded DNA template molecule in purple, we've now got 4 double-stranded DNA molecules of various lengths. Let's go through one more cycle of PCR. For the sake of simplicity for displaying this on my screen, let's just focus on what happens to these 2 DNA products in the next cycle. As you'll see in a second, these are the DNA strands that are going to give us the PCR product that we really care about in the end anyways. So now we're on to the 3rd cycle of this PCR reaction. It should be close to being old hat for you guys now, but what is the first step in that cycle? It's the denaturation step. The reaction will get heated up to 95 degrees, and the double-stranded DNA will be denatured and separated into single strands. Proceed to step 2, the annealing step. The temperature lowers to around 60 degrees so that your PCR primers can bind to the single-stranded DNA. Then comes the 3rd step, extension. DNA polymerase will make new strands off of your annealed primers. And now we finished 3 cycles of our PCR reaction. At this point, we now have the first copies of the double-stranded DNA product that we were really trying to make with PCR in the first place, which, remember, was to make a copy of a region of the DNA template that we were interested in, bounded on the 5' end of each strand by your primer sequences. PCR continues, though, after just the first 3 cycles that we worked through. Usually PCR is run for 30 to 35 cycles or more, and by the end, you'll have made 1 billion plus copies of the region of DNA you wanted to amplify. Alright, so with that review and walkthrough of PCR in mind, let's go back to the original practice problem. Let's begin with Part A. Read again through the problem and Part A's question. What information do they give us, and what are they asking for? Well, first of all, they give us a double-stranded sequence. Let's copy that here below so we can work with it. They also tell us that we want to amplify this sequence, so that basically means that this DNA sequence is the region of interest that we want to make 1 billion plus copies of by using PCR. The last bit they give us is a primer sequence. They tell us that primer number 1 you'll use has the sequence TTA-CGGGCAT. Thinking back to our PCR walkthrough earlier in the tutorial, we know that this must be one of a pair of PCR primers. We also know that primer number 1 here has to match the sequence at the 5' boundary of one of the 2 DNA strands in purple. By lining up the primer sequence with these 2 ends, we can see that primer number 1 matches our upper strand in this practice problem. Okay, those are our givens. Let's start walking through PCR. The first step is the denaturation step. We heat to near boiling and separate the double-stranded DNA. Next we'd have the annealing step, where primers bind. Primer number 1 will bind to its reverse complementary sequence, which you can see will be on the strand across from the one whose sequence it matches. Now we get to what Part A is actually asking. Part A wants to know, what are the first 5 nucleotides added to primer number 1 by DNA polymerase? DNA polymerase synthesizes a new strand in the extension step of PCR, starting by adding the first new nucleotide onto the 3' end of primer number 1. We can see that by complementary base pairing against the strand that the primer is annealed to, the first nucleotide after the primer will be a G, base pairing with a C. Then we can keep going. The next nucleotide added would be A, base pairing with T, then another A pairing with T, then a T base pairing with A, and finally a cytosine, or C, base pairing with guanine, G. You can figure out these first 5 nucleotides added by looking at the base pairing like this. But you may be thinking that there's a quicker way, and you're correct. Remember that primer number 1 sequence is identical to the 5' end of the other purple DNA strand, the upper strand. The new strand grown off of the primer number 1 here will thus have the same sequence as that of the upper strand, too. So when you're looking for the first 5 nucleotides added onto primer number 1, you could also get them as the next 5 nucleotides after the matching sequence in the upper strand. Either way you do it, the answer then for part A is going to be GAATC. Now let's work part B of the problem. Part B wants to know which of these 2 primers, primer number 2 or primer number 3, could be the missing primer out of your pair of PCR primers needed to amplify this given DNA sequence. You've been given the target DNA sequence. You also know one of the primers, primer number 1. In working part A, we saw that primer number 1 has the same sequence as the 5' end of the upper DNA strand. That means, recall from our earlier walkthrough, that the missing primer has to match the 5' end of the other DNA strand, the lower strand. So now our task is simple. Figure out which of our 2 primer options, primer number 2 or primer number 3, matches the 5' end of the lower strand. Let's paste the 2 primer sequences here for comparison. As you can see, they're kind of in the wrong orientation for us to easily compare them. Primer number 2 and primer number 3 are running 5' to 3', reading left to right, but the lower strand is running in the opposite direction. To make it easier to line them up with the lower strand and compare, what you'll need to do is rewrite the 2 primers in the other direction, or flip them around. So let me do that here for you. Okay, now take a second and see which primer lines up with the 5' end of the lower strand of the template DNA. After you compare them for a bit, you'll be able to see that it's primer number 3 that matches the outer 5' end of the lower DNA strand. So what that means is that our answer has to be primer number 3, and that primer number 3 is the other primer in part B that can work with primer number 1 to amplify our given DNA sequence by PCR. With that, we reach the end of this practice problem and also this tutorial for BIOS E1A. I hope that you found this helpful, and please remember, again, that if you have additional questions on the practice problems or course material, please do post them to the online discussion forums. Thank you all again very much for listening.